Let's talk about neurotransmitters. Neurotransmitters are awesome because they are the chemicals. We say that the nervous system is electrochemical. The electricity are things like the graded potentials and the action potentials. Chemicals are the neurotransmitters. There are something like at least 50 neurotransmitters, and there are even different categories, neuromodulators. I'm going to make you learn about a dozen of them, some of the main ones that have the main behavioral consequences. So remember, we have ligand-gated channels. Those are ion channels that are opened by chemicals, and specifically with the nervous system we're talking about channels that are opened by neurotransmitters. So three kinds of gated channels. Remember, the voltage gated, the mechanically gated, and the ligand gated. When we're talking about neurotransmitters, we're talking about ligand gated channels. All right, specific chemical stimulus. And in particular, what's going to happen here is that when a neurotransmitter binds to the receptor, and that's the term we use, it binds to the receptor, and then it opens the ion channel. All right. So neurotransmitters, we saw this at the neuromuscular junction. So this is not a completely new idea here. You've already been through this. We're just going to keep refining it. So one thing we didn't talk about at the neuromuscular junction, just because it was your first physiology ever and I was trying to keep it a little sort of under control. When the action potential comes down the axon, then goes down the telodendria, and finally gets down to the axon terminal, remember, it's been opening voltage-gated sodium and potassium channels all the way down. Well, at the axon terminal, there are voltage-gated calcium channels. And the voltage-gated calcium channels are opened by the action potential. They allow calcium through, and calcium is what causes exocytosis. Remember, that's the technical name for the release of neurotransmitters from the vesicles. All right. So the neurotransmitter binds to the postsynaptic receptor. Binding opens the ion channels. Influx of ions creates the graded potentials. And then we saw this at the neuromuscular junction. Remember, once we release those neurotransmitters, they're going to diffuse across the synaptic cleft, bind to the receptors, and the receptor, the channel, will open. But remember, if the neurotransmitters stay out there, they will keep binding and rebinding and so on. In other words, when they bind to the receptor, it only holds on to them for a tiny amount of time, and then it lets them go. And then they float around in the synaptic cleft, and then they can bind again. Remember, at the neuromuscular junction, I was saying that, if the neurotransmitter stayed out there, your muscles would contract for the rest of your life. And so we had to get them out of there. Do you remember what happened at the neuromuscular junction? How did we get acetylcholine out of the synapse? Yeah, we used acetylcholinesterase, an enzyme. So that's one strategy. Enzymatic degradation is the general term for that. It means that there are enzymes out there in the synaptic cleft that will tear the neurotransmitter to shreds. All right, and therefore it won't be able to keep binding to the receptor. But there's another strategy that's used in the nervous system, and that is reuptake. So literally, they're kind of like vacuum cleaners. They suck the neurotransmitters back in and recycle them. Makes sense. They get recycled either way. Even with enzymatic degradation, they get torn apart, but then they still end up ultimately getting recycled. Same way with reuptake. And in some cases, it's actually the axon terminal that does the reuptake. It can also be glial cells that do it as well. So we'll see that coming up. Here you see up in the top photomicrograph, you can see vesicles bursting open, releasing neurotransmitters into the synaptic cleft. All right. So it's what we've seen before there. Show you, I ain't making this shit up. See, there's actual photomicrographs down at the bottom. Look at all those vesicles. Yeah, it's really happening. Okay, graded potentials. We've been over this a few times. Let's go over it one more time. When the neurotransmitter binds to the receptor and opens the ion channel, the flow of ions creates electrical current. So remember, if a sodium ion flows through, that's positive electrical current. We call that an EPSP, excitatory postsynaptic potential, because it pushed the membrane towards threshold. Whereas if we opened a chloride channel, That created an inhibitory postsynaptic potential, an IPSP, because that made the membrane more negative. That pushed it away from threshold. So, therefore, the effect of a neurotransmitter can be either excitatory or inhibitory. An excitatory, depolarizing postsynaptic potential is called an EPSP. Depolarizing means making the membrane more positive, and that results from the opening of ligand-gated sodium channels. The postsynaptic cell becomes more positive or depolarized, and the postsynaptic cell is more likely to reach threshold. So you can see in the graph there on the left, we've got resting the membrane. Remember, what's the resting membrane potential? That was negative 70 millivolts. So if we open some sodium channels, we get an EPSP. That pushes it more positive. Then notice if we open some chloride channels, we get an IPSP. That makes it more negative. And look what can happen if we have a bunch of EPSPs. They may make the membrane so positive that it makes it all the way up to threshold. Remember what the value for threshold is? Negative 55 millivolts. And so in that case, that's what triggers the action potential. All right? So let's all review. Okay, so just putting all this together still, we have the EPSPs, we have the IPSPs, the hyperpolarizing postsynaptic potential. Hyperpolarizing, remember, means making the membrane more negative. That can result from the opening of the ligand-gated chloride or potassium. So remember, chloride is mostly out in the extracellular fluid, so its concentration gradient pushes it in. But because of the sodium-potassium pump, potassium is inside the membrane. Therefore, its concentration gradient pushes it out. So the moment we open a potassium channel, positively charged potassium leaves the inside of the membrane and flows outward. That means there's less positives inside. Less positives is the same as more negative. So EPSPs caused by sodium, IPSPs caused by either chloride or potassium. Postsynaptic cell becomes more negative or hyperpolarized. Postsynaptic cell less likely to reach threshold. I mean, we've got to get from negative 70 to negative 55. So if we have IPSPs, that's taking us to negative 71, negative 72, negative 73, negative 74. It's taking us the opposite direction. We're going away from threshold. And so both excitatory and inhibitory neurotransmitters are present in the CNS and the PNS. So what that means is that there's, in some cases, a neurotransmitter always, for example, opens sodium channels. In other cases, a neurotransmitter always opens chloride or potassium channels. So therefore, some neurotransmitters are always excitatory because they're always opening sodium channels, always creating EPSPs. And then vice versa. Some neurotransmitters always open chloride or potassium. We say that they're inhibitory neurotransmitters because they're always creating IPSPs. Now the thing is, though, the same neurotransmitter can be excitatory at some synapses and inhibitory at others. Acetylcholine is a good example. At some synapses, acetylcholine opens sodium channels and it creates EPSPs, therefore it has excitatory effects. At other synapses, acetylcholine opens chloride or potassium channels, therefore it has inhibitory effects. So you see, bottom line is, even though there are some neurotransmitters that always do one or the other, a lot of neurotransmitters can open either sodium or versus chloride or potassium. So at some synapses they're excitatory, at some synapses they're inhibitory. So you see, ultimately it's the receptor that determines whether it's excitatory or inhibitory, not so much the neurotransmitter. Yes, there are a few neurotransmitters for which they're always excitatory or inhibitory, but a lot of other neurotransmitters can be either or, just depending on the receptor. What kind of ion channel is the receptor, okay? Now, I've been kind of lying to you a little bit. Of course, you notice I've been doing that the entire semester. But we've talked about how neurotransmitters open ion channels, and in a lot of cases that's true, but they can do other things as well. So let's sort that out right now. So some receptors directly open ion channels when the neurotransmitters bind. These receptors generally bring about fast-acting changes. So in the upper left, you can see there, ligand would be acetylcholine in this case, for example. 2 acetylcholines bind to the receptor. It immediately opens the channel, and in this case both sodium and potassium are flowing through. This is a generalized cation channel, not just one or the other. We call that type of receptor ionotropic. So ion for ion. Tropic means change or grow. So ionotropic receptors rapidly, directly change ion concentrations, all right? And these are generally fast receptors, okay? In contrast, there are other receptors where the neurotransmitters do not directly open the ion channels. Instead, what happens is the neurotransmitter binds to the receptor and it activates another chemical. We call this, therefore, a second messenger system. So look down there in the lower right. We have the ligand, which would be the neurotransmitter. Notice it's binding to a G protein coupled receptor. So this is a receptor. It's still a receptor for neurotransmitters, but it doesn't directly open an ion channel. Instead, this receptor activates what's called a G protein. The G protein then in turn activates an enzyme, and then the enzyme goes about causing changes inside of the cell. All right, so we don't directly open an ion channel here. We activate a series of steps that bring about metabolic changes within the cell itself, okay? These channels, sometimes called metabotropic channels, because they change the metabolism of the cell. And we call these G protein coupled receptors GPCRs. So notice there are a few terms that all kind of get thrown together here. These receptors, by the way, are going to bring about slower but more long-lasting changes. So whereas the ionotropic channels directly open an ion channel, that allows ions through, causes immediate changes, but then the moment the receptor closes, the ions no longer flow through and the change stops. But these GPCRs, these second messenger systems, these metabotropic channels, see, all three of those terms get used. Second messenger system, metabotropic channel, GPCR. In this case, the neurotransmitter activates proteins, which activate enzymes. They change the cell's metabolism. These are generally much more profound, much more long-lasting changes, okay? Here you see again, over on the left-hand side, here you see an acetylcholine molecule binding to what's called a muscarinic receptor. It activates a G protein. Notice ultimately the G protein does open a potassium channel, but we didn't open the ion channel directly. So this is a GPCR. It's a delayed response. It takes longer. It doesn't immediately open the ion channel. All right? So it indirectly opens the ion channel. In other cases, like in the bottom there on the right, you can see that it goes down inside the cell, causes changes in gene transcription, receptor regulation. So these are more profound changes. So just to test your knowledge, that acetylcholine receptor, remember how we talked about it's more the receptor than it is the neurotransmitter? Let's take a look at this acetylcholine receptor. It's called a muscarinic acetylcholine receptor. We'll talk more about that later when we get to the fourth section of the class. Is that going to produce an EPSP or an IPSP? Do you know how to figure that out? You do. How do you figure that out? This channel is ultimately opening a potassium channel, and what happens when we open potassium channels? We get an IPSP, because the positively charged potassium leaves the inside of the cell, making the inside of the cell more negative. So we just saw the muscarinic acetylcholine receptor. Here's a different kind, the nicotinic acetylcholine receptor, and we'll be talking about these a lot during the fourth section of the class. So this is just a little bit of a preview. Just to explain what's going on with these channels, in this case this is an ionotropic channel, all right? The nicotinic channel is an ionotropic. Remember how a channel is really, the receptor is just a bunch of amino acids. It's a big giant protein that all kind of corkscrew through the cell membrane. Well, look in the bottom right. You can see that what happens here is that at rest, that channel, the subunits are so tightly packed together that even though there's a little bit of an opening, it's not a big enough opening for ions to get through. What happens is when the two acetylcholines bind to it, they cause a configurational change in the receptor. The receptor alters its three-dimensional structure, and now the opening becomes bigger and the ions can get through. So the only point of this slide was just to show you that. In fact, it's like we saw with calcium binding to troponin tropomyosin. It caused a conformational change in the troponin tropomyosin complex, and that moved it out of the way so the myosin head could grab the actin filament. It's the same thing here. This is a common theme in physiology. A lot of times, remember, proteins are what causing a lot of the activity inside of your body. They're the workforce inside of your body. So in many cases, proteins, when they bind with some sort of chemical stimulus, they undergo conformational changes, and that changes what the protein does, okay? So nicotinic ACh receptor is ionotropic, whereas the muscarinic we saw before was metabotropic. So let's do a little test your knowledge thing here. The opening of a ligand-gated potassium channel would cause a what, making it more or less likely for the membrane to reach threshold. So is a potassium channel going to cause an IPSP or an EPSP? And will that make it more or less likely for the membrane to reach threshold? Right, we just talked this through a moment ago. Potassium causes IPSPs, which hyperpolarizes the membrane, makes it more negative, that makes it less likely to reach threshold. We'll continue with more neurotransmitters in the next PowerPoint.